Hello, welcome to the video. In this video we're going to be explaining different types of white blood cell and clonal selection, clonal expansion and how white blood cells work. So firstly, as I'm sure you're aware, there are two types of white blood cell generally in the body. We've got phagocytes, we've got lymphocytes. So we need to know what they do, what their role is and what the different types are. So phagocytes, the two different types of phagocytes are neutrophils and we've got macrophages. So their basic job is to engulf and destroy pathogens by a process of phagocytosis. So that's what phagocytes do. So that involves them detecting the antigen on the surface of the pathogen, engulfing it, forming a phagosome, then a lysosome, which is enzymes contained within the white blood cell, fuses with the phagosome to form a phagolysosome, and the enzymes then break down the pathogen. So there's a slight difference between them. In terms of if you were given a picture and tried to tell between the neutrophils and the macrophages, you could tell the difference because neutrophils have what's called a multi-lobe nucleus. Their nucleus is kind of divided into sections, whereas macrophages have a more kind of traditional nucleus in the way that you'd have seen it before. The reason for that is because the multi-lobe nucleus allows the neutrophils to kind of move through blood vessels, move out of them into tissue fluid where they can fight off pathogens there. So that's the job of phagocytes. Another thing that can happen is macrophages, once they have broken down, digested the pathogen, they can then display the antigen on their surface. And that antigen can then be used to activate lymphocytes. So we can kind of say that phagocytes are non-specific. They can engulf and digest basically any type of pathogen. Whereas lymphocytes, there are two types. We've got T cells and B cells. And they have different jobs as well. So the T cells can be broken down into a few different types. T killer, T helper and T regulator. Now the job of a T killer cell is to detect any of our host cells, so any of the organism cells that are infected, because they'll be releasing distress signals with maybe molecules on the surface and releasing chemicals. The T killer cells are attracted to that. They can bind onto the surface of a cell that's infected and then they can release chemicals to destroy it. So that's the job of a T killer cell. T helper cells are basically involved in activating B cells, and T regulator cells can help regulate our immune response by reducing it once we've fought off the pathogen. So the job of B cells is to produce antibodies. So once a B cell is activated, it can become a plasma cell, and that cell will then release antibodies which can bind onto the antigens of pathogens, stopping them from functioning. Some B cells can also become B memory cells. Now the job of a memory cell is to remain in the bloodstream after infection. So if you're again infected by the same pathogen, you can very quickly produce antibodies to help fight it off. So you may have seen a graph that kind of looks something like this. With the x-axis basically meaning time, the y-axis meaning the concentration of antibodies in the blood. So we could say that at this point here, that could be the point of an initial infection. So if you're infected by a pathogen, the initial response isn't that great. You don't produce that many antibodies, which is why you display symptoms. However, if you then get infected at a later date, let's say here, you very rapidly produce antibodies, getting to a very high concentration in the blood. And what this does is it essentially fights off the pathogen before you display symptoms to the infection. Okay. So that is how that's what that graph means, and you've all experienced it. I'm sure that most of you watching this video will have had chickenpox at some point in your lifetime, but you haven't had it again. But the chances are that you've been exposed to the pathogen again, just that you haven't displayed symptoms. That's because the memory cells in your blood very quickly produce antibodies and they can fight it off. Okay, just one final word on clonal selection and clonal expansion. That refers essentially to these T and B cells. Now, in our body, in our blood, we have many different types of T and B cell, and they all will be able to produce different antibodies in the case of B cells, or they'll have different molecules on their surface in the case of T cells. Clonal selection is when essentially the correct T or B cell is selected. That will involve them recognising the antigen of the pathogen, and then, once it's been selected, the correct T or B cell will divide and produce many, many copies of itself to allow the body to fight off the infection. T cells, the reason they're called T cells is because they mature in the thymus. All of these cells are produced in the bone marrow, but T cells mature in the thymus, which is the gland kind of just above the heart, whereas B cells, they are produced in the bone marrow and they also mature in the bone marrow. Thank you very much for watching. If you have any questions, please leave them in the comments. If you have any requests, please do the same, and please follow me on Twitter at Rushcliffe Bio. Thank you very much.